Okay, let's see. All right, so next up we have Ben Americus. He got his start in salmon work aboard his father's commercial fishing boat in Prince William Sound, Alaska. In 2013, he moved south to attend Montana State University in Bozeman and helped with research on the rainbow trout gut microbiome. After getting his bachelor's degree, he joined Dr. Jerry Bartholomew's lab group at Oregon State University in Corvallis for grad school. At OSU, he was introduced to fish pathology and earned a master's degree with a thesis on myxozoans Ceratonova shasta and Myxobolus cerebralis. Now he's in his first year of his PhD at Oregon State and is applying omics tools to understand the weird and fantastic nature of myxos. So take it away, Ben.
Thanks, Nick, and thanks everyone for showing. I'm happy to be a part of this. Cool. There we go. Cool. Yep. So I'm out here in Oregon. Out here in Oregon, most of us study salmon diseases. Here we have some rainbow trout, and a lot of our salmon and trout around this area, they end up looking like this, kind of distended stomachs. You cut them open and you see these hemorrhagic intestines, and then you look at them under the scope and you see they have this parasite in them, this myxozoan parasite, Ceratonova shasta. And a lot of my work for my master's and for my PhD has been looking at these guys under the microscope. This C. shasta is kind of the bread and butter of Jerry's lab. It's our model myxozoan. But since we've all started to work from home, I've done a lot more computer work on C. shasta, looking into the omics side of it, and that's what I'll be talking about today. So there's a lot of interesting things about myxozoans. There's a great talk earlier in this series on Hennegaia in catfish, but I think in my mind the most interesting thing about them is that they belong to this phylum Cnidaria. So this includes things like corals and jellyfish and anemones, big macroscopic animals. And the thought here with the latest molecular clock analyses is that myxozoans diverged around 650 million years ago, sometimes 600, 650, kind of approximate. And since then, so that's this big branch here, way out to this myxozoa out here. Since then, they've reduced their genomes. They've evolved to become parasites. They've evolved to become microscopic. And just to put this in context, this is like 650 million years. It's like over 100 million years before fish emerged or before there's any animals out there with a backbone. So quite, you know, this divergence is pretty remarkable. So this has been known since like 1999. It was suspected before then, but kind of concluded with the sequence data. And since then, people have kind of been trying to rationalize the role of these myxozoans and what their characteristics are within the phylum Cnidaria, things like jellyfish, corals, and anemones. Kind of a unifying trait of all of these organisms is that they all have nematocysts, which are these stinging cells. Anybody that swam with a jellyfish knows what these are about. So a lot of my master's work, which wrapped up this last fall, was looking at comparing and contrasting myxozoan and cnidarian nematocysts and trying to exploit what we know for treatments. So we published a couple papers on this. The first one was a review paper comparing and contrasting the structure of the nematocysts, and we found that in both myxozoans and things like jellyfish, they're full of these ions, these little positive guys here. And then we did some wet lab experiments and found that if you put the Myxobolus cerebralis in high salt conditions, it interferes with its ability to infect fish. And this just came out in the latest issue of Parasitology, just like about a week ago. So I told this to my brother, who's a fisherman still, and his response was, so you learned that a freshwater animal doesn't like salt. So I guess since then, since October, I've really been kind of stepping up my research and looking for alternative methods to try to impress him more. So a lot of what I read about is the work of you all in fish pathology, but also these papers on cnidarian, just Cnidarian crazy cell biology. And a lot of my favorite papers that have come out in the last couple of years are about anemones, and specifically this one anemone, Nematostella vectensis, shown in the middle here. And the people are doing some really wild things with this anemone. They're making transgenic ones. They're causing the mRNA to glow. They're mapping out the different proteins in an entire organism. And then, yeah, they're doing some crazy, recently, kind of my latest interest is they're doing some really crazy electrochemical stuff with the nematocysts. So looking at all this research, I'm trying to kind of, I think, adapt some of this work with anemones into the work with myxozoans. And a lot of this work with Nematostella vectensis has been around this idea of venom. Like I said earlier, all cnidarians have these nematocysts. Most of them contain venom proteins. And so when you're stung by a jellyfish, what it's doing is using its nematocyst to inject a venomous protein into your muscle cells or into your blood. And these take a bunch of different forms. These little protein structures here, a bunch of different families, and some of them are shared between cnidarians and some are unique to individuals. So in kind of this is my first bioinformatic paper, and my first work in applying some of these tricks in anemones to work with myxozoa. We went after this question of, do myxozoans use venoms to infect fish? And this kind of took the form of 3 questions. Do they produce venom proteins? How do they use them? And then if they have them, where do they get them from? And a big kind of thing that tipped us off to this really early on was one of our collaborators, George Sisler out of Colorado, sent us these really nice photos of Myxalba cerebralis infecting baby whitefish. And you see this really crazy cratering. So this is the head of M. cerebralis here, and you see this crazy cratering effect. There's definitely some like proteinase activity or something happening here. And this looks a lot like what you would expect from a jellyfish sting. So I'm lucky in Jerry's lab, I got a lot of data to work with to approach this question. The first data set is this polar capsule proteome. So back in 2017, this is published a while ago. They isolated C. shasta myxospores, then used this lab on a chip technique to isolate nematocysts from them, or typically nematocysts are called polar capsules in myxozoans. And then used LCMS to get a list of proteins that these nematocysts are made up of. And then the other data set came around more recently. It's developed by another graduate student in Jerry's lab who's on this call, Damian Barrett. And what he did is extracted RNA from a time series infection. So he infected rainbow trout or steelhead with C. shasta, and then took RNA samples at 1, 7, 14, and 21 days. And then one of our collaborators developed these into a time series transcriptome. So the kind of interesting thing about this data set is it essentially tracks a healthy fish to a dying fish, a dead fish. At day 1, you don't see any pathology, but the thought is these parasites enter through the gills. Day 7, you don't see too much. This is the intestine here. But by day 14, 21, you see these nice folds of the intestine just turning into soup. And this is because of the parasite. Whether it's expressing proteases or what it's doing inside of there is still kind of unknown. So yeah, in trying to approach this question, I developed a bioinformatic pipeline. A lot of times when people say bioinformatic pipeline, I at least think of just a button, you press go, and then it spits out data at the end of it. And this is kind of that, but this pipeline that I made for this was also a lot of like hands-on, finicky kind of figuring things out. And I'll go into each of these categories, but kind of the goal of this process was to independently kind of answer each of these questions, find some venoms, and then look at how they're expressed, and then kind of try to figure out where they came from. So the first question, does C. shasta have these things? I worked on this with a postdoc in Jerry's lab, Nicole Hams, who's moved on to Fish and Wildlife. We kind of took a 2-pronged approach. I scoured the internet for everything that was annotated as venom and made this big database, big transcript and protein database. She used this other approach where she kind of uses program Fire2 to structurally model these proteins. And together we ended up with 28 venom proteins. And I should note, my approach is this is more typical for looking for interesting sequences. This structural modeling is a little bit newer, and what it relies on is the fact that with really divergent proteins, like proteins that belong to this organism that diverged 650 million years ago, they're probably not going to be very similar to something in a jellyfish, but the structure might be similar. So you can use this structural modeling to look for similar ones. And in hindsight, I think that her approach is a little bit better for looking for really divergent proteins than mine was, just because this thing's evolved so far out. Together she found 28 and I found 14, and she found all of my 14. So I think the answer to this first question is yes. We did find quite a few venom proteins that were either structurally similar or similar in sequence to the ones that we know exist in things like jellyfish and anemones. And these include things like proteases, isomerases, things that tear up proteins, things that bind proteins, things that clot blood or interfere with blood clotting. And unfortunately, there's some really nice pipelines that exist, just push but go button pipelines on the internet that were designed for anemones. And I was able to put these 28 through them and find 6 that I really think are high confident venoms. I think when we publish this or when we submit this, there'll be some pushback. There's several papers already that have said that myxozoans don't use venom. So I'm going to kind of pick the smallest battle here and just say we have 6 of really high confidence, not 28. So the next question is, how are these things, how does shasta use these? How are they expressed? So using this time series data set, we can look at expression over time. So red is high expressed, blue is low expression. Here's our 6 high confident venoms on the right. And so we have 1 day in the gills, 7, 14, 21 in the gut. And what you see here are 2 kind of different clusters of expression. These proteases are expressed really heavily in the gills and then in the gut out 7 days, and then they kind of die off later on and kind of kick back up at 21. These other ones, these inhibitors, this one that's related to blood clotting, it kind of kicks up over time and is really most heavily expressed at 21 days when this thing's turning into soup. So I think to answer this question, I think C. shasta is using them for 2 different purposes. Probably the first case where it's using all these proteases, that's for mobility. Getting from the gills to the gut takes a lot of bursting through different cell walls and kind of wiggling through cell junctions. And then at 21 days, essentially this thing is a factory for myxozoan parasites. So it's going to need to kind of use a more diverse set of transcripts. I get a lot of questions on these heat maps. So I think a better way to present this maybe is in these kind of pulse charts here. So you see proteases really heavily expressed at day 7, dying off at 14. And then these other types kind of increasing over time in the infection. So the last question, I think, so we know that they have, or we're pretty sure that they have these venoms. We see that they're expressed in these 2 different ways. So where do they come from? Fortunately, there's a lot of great proteomic data on NCBI from other cnidarians. And when I just put this together in a phylogenetic tree, I see that it really clusters by taxonomy here. We have all the corals together, anthozoas, the jellyfish and anemones grouped together, and then these weird myxozoans, they also group together. So Hennegaia, the catfish one, and then Myxobolus squamalis grouped together. But to really get at this, I think you have to expand your view to elsewhere in biology. So kind of an interesting thing about working on this project is I've seen a lot of like blast hits to things like snakes and urchins, other venomous organisms. And when you include these other venomous guys in the analysis, you see that C. shasta continues to group with these other cnidarian, with its relatives. So I think this led me to believe that I think these venoms came from other cnidarians. And kind of looking at this tree as a whole, you see that the echinoderms, these urchins kind of group together, the snakes group together, venomous snails and frogs are kind of independent. But essentially what you have here is vertebrates and then invertebrates. And this has been done by some other researchers, not with myxozoans, but with other venomous proteins. And the thought is that basically all animals have these proteins, but just for whatever reason, randomly, the selective pressures in life cause them to become venomous. There's little, this is the alignment file for this phylogeny. It's really crazy, really not very clean, but it comes together nicely. But what's happening here is that the selective pressures in these different environments cause these normally non-venomous proteins to be used as a weapon, which I think is pretty profound and pretty interesting, that this thing that is evolved in C. shasta to play this role has evolved similarly in something like a snake to be used as a weapon. So that's my talk. I'd like to thank definitely Damian Barrett for developing that really nice time series data set. The CGRB here at OSU for helping me with this computational work. The OWL and my fellow Bartholomew Lab members for good feedback. And my funding comes from this Binational Agricultural Fund with Israeli collaborators, and then more recently from the Bureau of Reclamation. But thanks for having me, and I'll take questions.
Awesome. Very interesting presentation, Ben. That's cool stuff. Go ahead and unmute yourself if you've got a question for him, or you can put it in the chat box and I'll read it. We've got plenty of time for Q&A.
Hey, Ben, this is Jan. How's it going?
Good. Hi, Jan.
Great job. That was an awesome presentation. Really interesting stuff.
Thank you.
Looks like with the venom, a lot of stuff that you're focusing on is like early in the infection with the actinospores getting into the fish.
Yep.
And I wonder if you can extrapolate some of that data to other organisms like Kudoa. Kudoa is in the muscle, and then during the late stages, when you have those myxospores, you get what they think are proteolytic enzymes that are released, and they lyse the muscle.
Totally.
That lysis. Do you think that that's venom related as well? And it doesn't seem like they're discharging their polar filaments at that point. Is there another mechanism that maybe they could use to release proteolytic enzymes or venoms at that stage?
Yeah, I think that's so just looking in things like jellyfish, the venoms are not only found in the nematocyst, sometimes they're expressed in cells of like the mouth and the stomach. So I think it's really possible that in something like Kudoa, these venoms are being expressed before the nematocysts even form, while it's just in its weird kind of metabolism stage. That'd be an interesting it'd be interesting to look at those proteins and see if they cluster with these like, I guess, these venom proteins, or if they're of course all of these organisms have a lot of proteases that aren't venoms that are just involved in metabolism.
Sure.
But yeah, I think even in our case, a lot of these venoms aren't expressed in the nematocyst. They're just expressed as this like blebbing cell.
Okay. Cool. Great job. Nice presentation.
Oh, thank you.
It's pretty quiet out there, so I'll ask another question since I'm really
Sure.
interested in myxozoans as well.
Yeah, thank you. No, for sure.
Just coming back to some of your introductory points, if these diverged 650 million years ago, it seems like fish hosts are a really common second host for myxozoans. So
Yes.
if they evolved 100 million years before fish have evolved, is there anything that supports that? Like, have we found any myxozoans that utilize only invertebrates as hosts and don't actually go into fish? I know that there's one myxozoan that can be directly transmitted between fish to fish, but is there anything that can complete its life cycle just within an invertebrate?
Yeah, I mean, I think that I know there's that really nice paper out published last year that shows that they co-evolved with annelids. So they were definitely, their ancestral state was with just replicating within annelids, and then they were introduced to fish. Yeah, I can't think of any on the top of my head that don't utilize fish. I mean, most of these myxozoans, people only know about their fish host, and the annelid host is still unknown.
Yeah, sure.
But yeah, if anybody else on here knows, I'd be interested in that answer as well.
Thanks.
Yeah, thank you.
Hey, Ben, great talk. This is Esteban. I was just wondering, and I know that you mentioned that, in the introduction that the other grad student was also doing, the RNA-seq throughout time. And I was just curious, as you follow the venom, and the myxozoan, I was just wondering, if there were evident changes, during the infection that took place on the fish as these, get expressed. Did any of them, may have, kind of like ticks work, that they release a number of different, virulence factors, that actually cause, a slight, immunosuppression and local immunosuppression, during infection to allow, infection to take place, So I was just wondering if you had looked into that or if that is just part of another other work.
Yeah, I think so I've only been handling the myxozoan side of the expression, but I have heard a lot of good talks by this guy Damien Barrett, and it does seem like a big part of the virulence with C. shasta is a big part of why fish die is because they're immunosuppressed, because they don't respond in time due to the parasite. So yeah, one of these venoms, I think, of high confidence, it's an inhibitor. I think I have to go into more check, but yeah, I wouldn't be surprised if some of this immunosuppression that you see, this reduced inflammation, reduced signaling that you see in fish is from these venom proteins.
And so you mentioned this is C. shasta, and you mentioned you presented a very nice figure of the electron microscope of M. cerebralis, into the trout, and I know that it has been shown, that a lot of the myxozoans, really enter, through the skin, at least, M. cerebralis. So I was just curious, if you guys, are looking into that for M. cerebralis and other myxozoans. Again, Jan proposed, the kudoa. I'm just curious, of the M. cerebralis, just, because it has been shown, that the skin, it's is the major portal of entry. So I'm just curious, if you guys have looked into that.
Yeah, that's actually, that's the next proteomic data set we have queued up, is these M. cerebralis actinospores. There's this paper we worked on with George that accompanied that. I'll go back to that SEM that accompanied this SEM that shows just the fish these little baby whitefish were dying just from the infection, not from any buildup of parasites. I guess that's something I'm really interested in. I think that it would make sense for these venoms to be causing this damage here and to be killing these little baby fish. I guess stay tuned. I think this is something we are working on, looking at these parasites in MC transcriptomes and proteomes.
Yeah, and if you go back, to the C. shasta, the last question, to the C. shasta table that you show, the histo figures, with the expression of the myxozoan. So can you go back to that slide?
Is that time series? So this.
Yeah, this one. Yeah. So, early on, I guess, like within one day, your challenges, with C. shasta, they are by immersion, right?
Yep.
So, do you see in the histological figures, on one day, I know that you can see, like, the parasites, but have you guys been seeing, like, changes in the gut mucosa, that could be associated with these, I guess, going back, to my same question, with these inhibitors, that you are showing in here, is the C. shasta causing any changes in the first, 24, 48 hours at the level of the gut mucosa, like, any type of inflammation or damage as it penetrates?
Yeah, I think there's this is Damien's finding, not my own, but there was no response at one day. It's like the fish, for these parasites that eventually killed the fish, there was almost no detection. I don't think there's any detection at one day post exposure. So it's like it seems like they almost catch them off guard. I've seen those nice histology shots of the C. shasta. Damien's raised his hand here. He might be able to answer this better than me. I'll go for it.
You can just unmute yourself, Damien, if you want. Oh, he's got no mic.
Oh, I see.
All right, he put something in the chat box here. It says there was, all right. Sorry, there's these security features where people can't chat with everybody. There you go. You can try it now, Damien. All right, did you see that?
Yep.
Okay.
Yeah, so Damien says that there is immunosuppression of the interferon gamma response in the gills, possibly of the Th17 receptor later on in the infection. So I think that gets on the suppression. And then, yep, Damien says no transcriptome response at days, I think 1 and 7 is what he means there. But yeah, it's, I think just 7. Okay. So yeah, it's like this parasite almost. My understanding is it kind of sneaks its way in until it's causing all of this damage, and then it's too late for the fish to respond.
That's very cool. Thank you.
Yeah. No, thank you.
All right, so we're out of time already today. I thank you very much, Ben. A virtual round of applause for you and Heather for the great talks this afternoon. We have a double feature next Friday from the University of Georgia, so be sure to check out the website for the Zoom link for that. I've also put in the chat box compliments of Bart at Michigan State University, a webinar that's going to be on Monday, hosted by WAVMA, Infectious Diseases of Farmed Marine Fish. So if you're piling up on webinars, there's another one for you. Until next week, everybody have a good one.